Okay, let's start discussing chapter number 17, and in this video I'm going to discuss about the next step, which is translation. So okay, so after mRNA is processed, it goes to the cytoplasm, and in the cytoplasm are ribosomes, and there the next step takes place. So basically, to understand translation, we need to first of all understand few components of the translation machinery, which involves tRNA, which is transfer RNA. We also need to know about ribosomes. So we will first of all talk about transfer RNA, then we talk about ribosomes, and then we will talk about the process of translation. So basically, transfer RNA, as the name itself says, transfer. So it transfers, what are the transfers? It transfers the amino acids. So the function of transfer RNA is to transfer amino acid. And how does transfer RNA transfers amino acid? This is let me show you the structure of transfer RNA. So this is the transfer RNA. So first of all, it's RNA, so it's single stranded. This is the 3' side of the transfer RNA, and this is the 5'. So this side is the side, 3' side, 3' end of the transfer RNA is that end where the amino acid is attached. And here is an anticodon. So we just talked about mRNA has codons. So transfer RNA has an anticodon. So anticodon is complementary to the codon. So this anticodon recognizes the codon on mRNA. Okay, so this is the structure of transfer RNA. It is basically looped here. You can see it's all because, but remember that transfer RNA is single stranded. It has just got looped here, and that this looping of the transfer RNA is because of the hydrogen bonds which form between the complementary bases in the same strand. So in some of the books you will see the structure of transfer RNA like this, or it is written, it's shown like this. So basically there are two parts of the transfer RNA. On the 3' end, amino acid is attached, and on the other side is anticodon. So transfer RNA transfers amino acid to the growing polypeptide chain in a ribosome. So this is the translation machinery. Basically, this is the ribosome. So ribosomes have two subunits: smaller subunit, large subunit, and these are the transfer RNAs, and this is the mRNA. And we are going to talk about them in detail. Now, the transfer RNA, as it transfers the amino acids, those amino acids are attached to the transfer RNA with the help of the enzyme called amino acyl tRNA synthetase. So with the help of this enzyme, the amino acids are attached to the transfer RNA. So you can see this is the enzyme, okay, and the amino acid goes and binds here, and transfer RNA goes and binds there. You can see how the shape of the active site is complementary for the amino acid. So the amino acid binds here, transfer RNA binds here, and with the help of ATP, the transfer RNA, the amino acid is attached to the transfer RNA. So when the transfer RNA has an amino acid, then that transfer RNA is called a charged transfer RNA. When there is no amino acid, then it is a uncharged transfer RNA. So remember that, because this terminology is going to come forward. If the transfer RNA has an amino acid attached to it, it is called a charged transfer RNA. If there is no amino acid attached to the transfer RNA, it is uncharged transfer RNA. So uncharged transfer RNA becomes charge transfer RNA with the help of the enzyme. And for different amino acids, there are different enzymes. Okay, before we go and talk about ribosomes, something important I would like to tell you is about Biol effect. Now, what is Biol effect? So, we just talked about transfer RNA. There are 45 transfer RNAs. Okay, there are 45 transfer RNAs and there are 20 amino acids, right? And there are how many codons? 61 codons, right? So there are 45 transfer RNAs, 20 amino acids, and for 20 amino acids there are 61 codons. So transfer RNA, as we just discussed, it has a anticodon, right? On this side is the anticodon. This is the anticodon. And this anticodon goes and binds or recognizes the codon. So anticodon is complementary to the codon. The codon on mRNA is okay. So now there are 45 transfer RNAs. There are 45 transfer RNAs and there are 61 codons. Some of the transfer RNAs, this is what is called the Biol effect. Now, how one transfer RNA can recognize more than one codon? The reason is the third ribonucleotide in some of the transfer RNAs is modified and it can recognize actually more than one nitrogenous base. Like G usually recognizes C, but there is some modification in some of the nucleotides in the third ribonucleotide in the transfer RNA that modified nucleotide can recognize, if it is G, it can recognize C also, it can recognize A also. So this effect is called Biol effect. So flexible pairing at the third base of the codon is called Biol and allows some transfer RNAs to bind to more than one codon. Okay, so that is what Biol effect is. There are 45 transfer RNAs, 61 codons. So some of the transfer RNAs can recognize more than one codon. So how is it possible? Because at the third base in the anticodon is modified and it can recognize more than one nitrogenous base or nucleotide in the codon. So it can, the third one is modified and because of that it can recognize more than one base in the, if it is G, it is C as it does normally, but also it is A, it can recognize T. So this is called Biol effect. Okay, now let's talk about ribosomes. Okay, ribosomes, basically you need to know that ribosomes, they have two subunits. One is the small subunit, another is the large subunit. On the smaller subunit, it has a mRNA binding site. On the larger subunit, there are three sites. There is a P site, A site and the E site. E site is the exit site. A site is a site where the new transfer RNA enters. And P site is a site on the ribosome where there is a transfer RNA on which the growing polypeptide chain is there. Okay. So this is the basic structure of the ribosome you need to know. And yeah, the ribosomes in the prokaryotes and eukaryotes, they are different. And there are some antibiotics which are against prokaryotic ribosomes. And it is, here is the significance that the ribosomes in prokaryotes and eukaryotes are different because, as I said, there are some antibiotics which target prokaryotic ribosomes. If the ribosomes in both the prokaryotes and eukaryotes were same, then these medicines were not possible because then these antibiotics would have attacked the eukaryotic ribosomes also and they would have affected the eukaryotic cells also. So then those medicines would not have worked. So this is the significance of the ribosomes being different in prokaryotic and the eukaryotic cells. So this is the smaller subunit of the ribosome, and here is the mRNA binding site, and this is the larger subunit of ribosome. Here is A site, P site and E site. E site is a site on the larger subunit where the transfer RNA can exit. Here is the site for here is a site on the ribosome, larger subunit of ribosome, where the transfer RNA will be there, on which the growing polypeptide chain is there. And A site on the ribosome, on the larger subunit of the ribosome, is that site where the new transfer RNA will enter and it will bring the new amino acid. Okay, that's the basic structure of the ribosome, the smaller subunit and the larger subunit. And you can see here, this is the mRNA and it binds on the smaller subunit. This is the larger subunit, and here are the transfer RNAs, and we are going to discuss in detail. Okay, there are three stages of translation. First is initiation, then elongation and termination. And every stage requires some protein factors, and energy is also required, of course. Okay, so let me first of all talk to you about the initiation step. Now, as the mRNA comes out of the nucleus, it is fully formed, matured form of mRNA. It comes and it binds to the smaller subunit of the mRNA, of the ribosome. Now, as I said, ribosomes have two subunits. These two subunits of the ribosomes are not together all the time. They are separate, okay? They associate only during the process of translation. So you can see here, this mRNA binds on the smaller subunit of the ribosome. Now the ribosome, the smaller subunit of the ribosome, it actually walks on the mRNA. It actually walks on the mRNA, and it will keep on walking till it finds the start codon, which is AUG. Okay. Once it finds the AUG, at this point, the transfer RNA with the amino acid methionine is recruited here. Okay. And at this point, the larger subunit of the ribosome also attaches, and that results in the formation of translation initiation complex. I would like to tell you something here, that as you know that start codon is AUG. So the anticodon on the transfer RNA is UAC. So this is the formation of the initiation complex. After that is elongation. Now what happens in elongation? Let's study from here. Now remember that, so when the important thing I would like to tell you here is, when the transfer RNA with methionine is recruited, it comes and binds on the P site. A site has to be always free because the new transfer RNA has to enter, and this new transfer RNA will bring the new amino acid. And which transfer RNA is going to come here and bind? It all depends on the codon in the mRNA. Just to give you example, suppose the next codon is GGC. I'm talking about codon. Codon is on mRNA. If the next codon is GGC, now the transfer RNA which will be recruited, suppose GGC is a codon which codes for the amino acid serine. Now the transfer RNA with the anticodon CCG will be recruited here, and it will bring the transfer RNA serine. So if it is GGC, so the transfer RNA will be here with CCG and it will be having serine. Okay. So once there are two transfer RNAs here, one here, suppose here is a transfer RNA with serine, here is with methionine. Now next step in elongation will be this amino acid will break its bond. This amino acid will break its bond with this transfer RNA, and then when this amino acid breaks its bond with this transfer RNA becomes uncharged, and then it translocates from P site to the E site. It comes here. the uncharged transfer RNA moves from P site to E site. It comes here. And then this amino acid, it forms a bond with the transfer RNA which is here. It forms a bond with the amino acid which is attached to the transfer RNA which is here. Suppose here is a transfer RNA with serine. This methionine is going to make its bond with serine. Okay, so here will be a transfer RNA with methionine, with serine, and this transfer RNA which is uncharged, it moves from this side to this side, and this transfer RNA which has a methionine and serine moves from A site to P site. This is all called translocation. Okay, because A site has to be always kept free, because this ribosome will walk on the mRNA. It will keep on reading the codons and it will keep on recruiting the new transfer RNAs. So once the translocation has taken place and the uncharged transfer RNA has reached here, from this place it will then go to, it will just leave the ribosome. That's why it's called the exit site. Okay, so basically what will happen is, on the P site there will be a transfer RNA which will have the chain of amino acids, Okay, so that's why it's a P site, because here will be a transfer RNA which will be having a growing chain of amino acid. This site will have the transfer RNA which will have a new transfer RNA with the amino acid, and this is a place from where the uncharged transfer RNA will leave the ribosome. So the new transfer RNA will be coming here. Okay, and whatever the amino acid is here, or if this will have actually 4 or 5 amino acids, or the growing chain of polypeptide, growing polypeptide chain will be here. This transfer RNA will break its bond with this growing polypeptide chain, and this will become uncharged. It will move from here to here, and this growing chain of polypeptide will make its bond. It's going to make its bond with the new transfer RNA, with the amino acid which is there on this transfer RNA, and this transfer RNA which will be having a growing chain will move from A site to P site, because A site has to be always kept empty. So this is elongation. Let me explain you more from here. You can see here what is happening here. This is E site, this is P site. Here the growing chain is of the this is the growing chain of polypeptide is here on this transfer RNA. This is the new transfer RNA which is coming. Okay, it will come here and bind here. Then this growing chain of the polypeptide, this growing chain of these amino acids, it is going to break its bond with this transfer RNA. This transfer RNA is going to become uncharged, and this growing chain is going to make its bond with this amino acid. Okay, now you can see here how the break is bonding, how the bond is breaking, and how this chain is making its bond with this amino acid, and this transfer RNA is becoming uncharged. So when this transfer RNA becomes uncharged, it moves from here to here. Okay, it moves from P site to E site, and from E site it exits the ribosome, and this transfer RNA which is having this growing chain is going to move from A site to P site, and the A site will be kept empty, because then the next transfer RNA will be coming and will be bringing new amino acid. So this thing will keep on going. This is elongation. This will keep on going till there is a stop codon on the mRNA, and that is the termination of translation. So when there is a stop codon, when there is a stop codon on the mRNA, at this point there is not a recruitment of the transfer RNA here in the A site, but instead there is the recruitment of the release factor. Now this release factor is a protein. It comes and binds here, and it helps in a hydrolysis reaction, and as a result of that, the bond between this transfer RNA and this growing polypeptide chain breaks, and as a result of that, this polypeptide chain becomes free. This transfer RNA becomes uncharged, and it moves from P site to E site, and from E site it is going to go out of the ribosome, and after that the both subunits of the ribosomes, they are also going to get disassociated. So the polypeptide chain is formed. Remember that this is a polypeptide chain. This is not a protein. So if you go back and check your chapter on biomolecules, you need to know what's the difference between polypeptide chain and protein. So once the polypeptide chain is formed, the two subunits of the ribosomes, they separate, mRNA separates, the release factor goes away, everything gets disassociated. After polypeptide chain is formed, there are many post-translational modifications. Which takes place to the polypeptide chain, and then eventually it forms it folds. Some of the post-translation changes takes place, like some of the proteins gets glycosylated, which means the addition of the sugar chains. So many things takes place after the translation. They are called post-translation modifications, and then the protein folds, further folds, and attains its three-dimensional structure. Now, what is polyribosome? Polyribosome is when many ribosomes, they are together translating the mRNA. So you can see here this is the mRNA, and you can see how many ribosomes have attached to the mRNA, and they are all helping in the translation process. So the ribosome which has reached here, it has already made the complete polypeptide. The ribosome which has reached here, it has made this much polypeptide. The ribosome which has reached here, it has made this much polypeptide. Which is just starting has made this much polypeptide. So polyribosomes are found both in prokaryotes and eukaryotes, and they help in increasing the efficiency of the protein expression. So this is all we have discussed. This is just a summary. You can go over them. And the last topic I would like to talk in this chapter is mutations. Now, what are mutations? Mutation is a sudden change or it's a random change in the DNA. Okay, so mutations are changes in the genetic information of a cell. Now, there are some, we are going to talk about different kinds of mutations. Point mutations are those mutations which are just in one nucleotide. Okay, so this is a typical example of a point mutation which takes place in sickle cell anemia. So this is the wild type beta globin protein. So this is the sequence in the wild type beta globin DNA, and this is the mutant. So in the wild type beta globin, so you know that hemoglobin is a protein which is having 4 subunits, 2 alpha chains and 2 beta chains. So there is a mutation in the beta chain. So this is the wild type. The sequence is CTC. In the mutant, it is CAC. And as a result of that, there is a change in mRNA. In the mRNA, it is GAG, and the mRNA in sickle cell anemia is sickle cell anemia is GUG. And as a result of that change in mRNA, there is a change in amino acid. In the normal hemoglobin, the amino acid is glutamate, which is charged amino acid, and in the diseased state, it is valine. Valine is non-polar amino acid. So this is a typical example of point mutation, and it causes sickle cell anemia. Now, the mutations, they can be substitution mutations, or they can be insertions or deletions. So substitution mutations are when one nucleotide is substituted for another. Suppose it's A, it becomes G. If it is G, it becomes T. So that's substitution. Deletions and insertions means if there is a deletion or the insertion of some of the nucleotides. So insertions and deletions are more dangerous mutations because they can totally change the reading frame, and they're called frame shift mutations. Okay, some of the mutations are called silent mutations, which means they do not cause any effect. The reason is if there is a mutation and it does not affect, why it doesn't affect? Because for some of the amino acids, there are more than one codon. Suppose there are there is a codon called, suppose it's GGC, it codes for serine, and there's another codon GGG, it's also code for serine. Suppose because of a mutation, the codon has from GGC, it has become GGG, it's going to code for the same amino acid. So these kind of mutations are called silent mutations. Missense mutations are those mutations which change the amino acid. Nonsense mutations, which actually, it's a kind of a mutation because of which the codon becomes stop codon. Okay, because of a mutation, the codon becomes a stop codon and it stops the process of translation. Those kind of mutations are called nonsense mutations. So let's and what the for the mutations can be caused by physical or the chemical agents, and they're called the mutagens. So chemical mutagens can be many chemicals can cause mutations, and the chemicals which cause mutations cause cancer, they're called carcinogens. Okay, now this shows you some of the examples of the mutations and the effects. Now this is the wild type gene, and this forms mRNA, and this is the sequence of amino acids in the protein. This is the primary structure of the protein. So let's see, if suppose there is some mutation takes place, substitution mutation takes place, and instead of G there is A instead of G. So you can see in the wild type there is G, and if the substitution takes place, instead of G there is A, instead of G there is A, as because of that in the mRNA there is U instead of C. Because of this change, there is a change in mRNA, and as a result of that, so this is GGU now. Earlier it was GGC. GGC codes for glycine. Now it has become GGU. It also codes for glycine. So this kind of mutation is called silent mutation. Now here it is T instead of C. In the wild type it is C. Okay. In the wild type it is C, but here it is T. So when there is T instead of C, in the mRNA there is A instead of G. Okay. And as a result of that, the amino acid will be serine instead of glycine. So these kind of mutations are called missense mutations. Okay, here you can see A instead of T. So instead of T, if it is A here, in the mRNA it is going to be UAG, and UAG is a stop codon, and these kind of mutations are called nonsense mutations. Here, suppose there is one extra A added. This is insertion. As a result of that, in mRNA there will be extra U, and as a result of that this codon becomes UAA, so it's a stop codon. Now suppose here A is missing, and as a result of that in mRNA U is missing, and as a result of that what happens is, if U is missing, okay, if U is missing, then the next codon is UUG. Okay, next codon is UUG. In wild type the codon is UUU, it codes for phenylalanine. Now the codon is, instead of UUU, it is UUG. It is UUG. When it is UUG, the amino acid is leucine. And after that, all the amino acids are going to be different. So because of this, A missing here, there's U missing here. After this, the whole reading frame changes. So these kind of mutations are called frame shift mutations. Suppose here this full codon, these three nucleotides are missing. TTC is missing, and as a result of that, AAG is going to be missing. So AAG codes for lysine. So here, as a result of that, one complete amino acid will be missing. So this is basically all about your mutations. So that's all about this chapter.